started with the plasma membrane, that is the outermost covering or the, let's say, outermost boundary of the cell. Then we move on to sort of the control center of the cell. There is a nucleus, right? Then what did we talk about? After nucleus, we talk about immediately after the nucleus, what do you have? You have the endoplasmic reticulum, right? Then attached to one of the endoplasmic reticulums, we talked about ribosomes. Did we talk about ribosomes? Did we talk about ribosomes? Or do we have to start Golgi today? Yeah. Did we talk about ribosomes? No. So, yeah, I mean, also in the process of talking about ribosomes in your ribosomes and this protein synthesizing structures such as your rough endoplasmic reticulum and ribosomes and also your nucleus. So I'm in the process of discussing those in the 2025 class as well. So in the 2025, we are now talking about molecular biology, right? So molecular biology in detail, you have to learn about the internal structure of the ribosome, right? And how from the nuclear pores, from inside the nucleus, how the mRNA comes out and comes into the ribosome, and how from the information in the mRNA you make proteins, right? So that is called transcription and translation process. So you have to learn about that in so much detail under molecular, right? With all the different enzymes involved and all the different steps, right? So for now, what are we learning is we are just going through the different parts of the cell that are involved, right? So before going to the deeper parts, you have to have the basic idea now. So that's what we are doing in these early sessions, right? Okay, uh, everyone take out your the resource, right? Do we have to start from Golgi today? Anyone? Do we have to start from Golgi? Yeah, no? We didn't talk about ribosomes. Ribosomes comes. No, we have discussed about ribosomes. Yeah, we have discussed, right? So we have to start with Golgi apparatus. So you have to be able to draw a rough diagram of Golgi with a few labelings, right? A rough diagram of Golgi with a few labels, right? So I mean, you won't be asked to draw in the A-level paper, but it's a common question in the term tests. Okay, so get the ability to draw a simple Golgi. Okay, it's like this, no? So the Golgi is closely associated with your smooth endoplasmic reticulum, and also, so you in here in the center you have the nucleus, no? Nucleus has two. The nuclear envelope has two layers, right? Inner membrane and outer membrane. So immediately sort of attached to the outer membrane, you have the rough. Sorry, give me 1 minute. Right. Sorry about that. Still being recorded. Yeah. So immediately continuing from your outer envelope or the outer membrane of your nucleus, you have rough endoplasmic reticulum, right? How do you define? How do you sort of distinguish it from the all the ribosomes attached, right? So from sort of then sort of continuing from the rough endoplasmic reticulum, what do you have? Because the rough endoplasmic reticulum is like flattened, right? It's like flattened layers and layers of membrane. So continuing from there, you have like these tubular structures. That is your what? If it's tubular, if it's rough, it's smooth endoplasmic reticulum, right? Smooth membranes, smooth and tubular. That is your smooth endoplasmic reticulum. And then very close to the smooth endoplasmic reticulum, where all the vesicles are coming, right? You have what? Your Golgi, right? You draw your Golgi sort of similar to rough endoplasmic reticulum, actually. It's like a flattened stack of membranes with two sides, okay? It's like a stack of membranes with two sides, okay? It has one side to receive the vesicles that are coming, from the endoplasmic reticulum, right? Receive the vesicles. So it has like imagine it as a post office, okay? So imagine it as a post office. So it has a receiving side to receive parcels, like for delivery, it's receiving parcels from one counter, right? So it has to receive parcels, and inside the post office, they will be sort of sorted, right? Sometimes the packages will be opened and to see what is inside. Then sometimes they will be again repackaged to send to different locations, and right. So they will be sort of processed, like it's similar to what happens in a post office. So these vesicles and their contents will travel through the Golgi, right? Through the different membrane systems of the Golgi. And from one end, they will sort of be released, right? So these newly modified vesicles go to different places in the cell and even outside the cell to perform different functions. Okay, so what you need to know is you have to know that this receiving phase where they sort of parcel baraganta, right, where they sort of receive the sort of the vesicles is called cis phase, right? The cis phase, right, where they sort of accept the incoming vesicle, cis phase, and where the vesicles are going out of the Golgi is called the trans phase. Trans phase. Okay. And these stacks and stacks of membranes of the Golgi is called cistern. Okay, cistern. Okay, and inside you have this. It's hollow. Inside these tubes, it's hollow. So you call it the lumen. So the interior of the Golgi is called the lumen. Right. This is basically it. Right. So about Golgi, what do you need to know? You have to define it. It's a stack of flattened sacs or cisternae. That's how you say it, right? It's a stack. Again, it has been stacked of. It's a stack of membranes, right? And we call the different sort of the layers of the stack as cisternae. Okay. And it has two phases, as the inner cis phase and the outer trans phase, right? The cis phase is located near the endoplasmic reticulum to receive incoming vesicles, and the trans phase gives rise to outgoing, incoming, outgoing. Okay. Golgi apparatus is abundant in secretory cells. So that is obvious, no? If you are secreting something, that means they will be packaged in these vesicles, membrane-bound sort of parcels. And these parcels, if the cell is producing a lot of these parcels, then you have to have a lot of Golgi apparatus. Okay. So that is the anatomy. That's all you need to know about Golgi, right? I'll show you some diagrams with the screen share. Any questions? Any questions? So you can see my screen, right? Yeah. That's the tube now. So just a brief idea. So this is what I was talking about. This is how the internal sort of the structure of a ribosome looks like. It's much more complicated than you think, right? So just have an idea. You'll learn about this in detail. This is the mRNA going through the ribosome, right? This is the small subunit. This is the big subunit. These are what? Anybody? What are these? This stuff. It's labeled. These are tRNA, okay? Transfer RNA, bringing amino acids towards the ribosome. Anybody know how many types of tRNAs do we have? How many different types of tRNAs do we have? Transfer RNAs. How many transfer RNAs do we have? Types of transfer RNA. I told you this when we are doing nucleic acids. How many tRNAs do we have? What is the function of tRNA? To transport amino acids. Right? To do this, how many amino acids are involved in protein synthesis? How many amino acids do we need to make proteins? There are so many amino acids in the nature, right? There are so many amino acids, but out of them, only how many contribute to make proteins, right? Only 20. So there's one dedicated transfer RNA for every amino acid, right? For example, if you think about glycine, there is a transfer RNA, tRNA, that is transporting only glycine, right? එයා transport කරන glycine විතරයි. එයා නිකන් lysine, histidine එක එක්කෙනත් එක්ක set වෙන්න යන්නේ. So the tRNAs are very faithful. The tRNAs are very faithful to their amino acids. So to transport the 20 different types of amino acids, you have 20 different types of tRNAs. That is something additional, right? What are we talking about? So this is the tRNA structure. I hope you remember, right? With the anticodon. Right. Anyway. Yeah, nothing for you to be interested right now. Let's go into our lesson today, that is cellular basis. And yes, in this diagram, in the tube, you can see there's a the transmission electron microscope image is also there of your Golgi apparatus. You can see this is the cis phase, right? So step number 1, the vesicles are coming from endoplasmic reticulum and fuse with the Golgi, and their internal content is released into the Golgi, right? It's like a delivery van coming into the post office and unloading the things. It can be proteins, it can be amino acids, right? It can be DNA or anything, right? That is sort of unloaded into the Golgi, and new vesicles are formed and they are sort of like go out of the Golgi from the trans phase. Okay, that is all basically all you need to know. So you can see Golgi, this is the stack. It's a stack of cisternae, and these are the different vesicles that are going out. Okay, right. So what do they say about functions of Golgi? What do they say about functions of Golgi in the resource? Collecting, modifying, packaging, and distribution of materials, protein and membrane lipids. Yes, that is an important function. They collect, modify, package, and distribute. Please remember those 4 terms. Okay. Mainly proteins and membrane lipids. Why membrane lipids? From where do you get membrane lipids? Who is making membrane lipids? Who is the membrane factory? Who is the membrane factory? These are MCQ points. Okay. Who is the membrane factory? Yes. Your rough endoplasmic reticulum is your membrane factory. So membrane lipids that are produced by your rough endoplasmic reticulum will also be received by the Golgi. Okay. Manufacturing cellulose and non-cellulose cell wall components such as pectin. No. Pectin. Pectin, you know pectin. Pectin is what? Polysaccharide. What is the monomer? What is the monomer of pectin? Monomer of pectin. Pectin you can find in the middle lamella. Middle lamella of cells. What is the monomer of pectin? Yes, galacturonic acid. So pectin, I have told you this, right? If you draw two plant cells like this, pectin is sort of the cementing substance, right? So this is two plant cells, right? So the green color ones are the cell walls. So in between two plant cells, sort of like a cement, right? In between bricks, when you're building a wall, you put cement between bricks. So your pectin is like that, sort of the cement between cells, right? Pectin. So manufacturing cellulose and non-cellulose cell wall components. So they make cellulose as well as there are other things in the cell wall, such as your pectin. They are also synthesized in the Golgi and produce lysosomes. We just have to talk about lysosomes immediately after. What is I have written here? We'll come to this concept of the cell plate. Cell plate, I'll mention when you're doing cell division. Okay, no need right now, right? A cell plate is the cell plate originates from the Golgi. For now, don't think about that, right? Any questions? Any questions about Golgi? Right? I'll give you questions to do. So, no, right? Any doubts? No, right? We can move on to lysosomes. Lysosomes, about lysosomes, we can make a note here, right? Lysosomes. There's some space here, no? So if you please, since you don't have the tute at hand right now, take down on a different paper or something, and when you have the tute, please complete it. And I want to see completed tutes, right? Send me photos, right? The completed tute, right? Okay. Then we move on to lysosomes. They are so important, lysosomes, right? What is the definition? They are single membrane bounded vesicles contributing to digestive activity. Okay. So inside the cell, there are so many things to digest, right? There are so many things to digest. These things can be from within, these digesting things, the things that are undergoing digestion can be from inside the cell, or they can come into the cell from outside. Do you get it? Right? So what they digest can be something that is already inside the cell, or that is coming from outside. So depending on that, we have several types of what you call as digestive activity. Okay, several types of digestive activity. I'll show it from your reference book, that is the Campbell. So lysosomes. This is one of the functions done by lysosomes. You have two functions done by lysosomes here. Main thing is, one of the most common thing is phagocytosis. Phagocytosis. So simply put, phagocytosis is what? The cell is eating. The cell is eating. So if you are eating, that means your food has to come from outside. So phagocytosis happens when the cell takes in food particles. When the cell takes in food particles. So it's like this. This is a food particle that is outside the cell. And through the plasma membrane, that food particle will be taken in. Okay, through the plasma membrane, this food particle will be taken in. Okay. So one of the best example is your white blood cells. This is a white blood cell, right? It's a not, I mean, it's a macrophage, right? It's one of the biggest, what do you say? Uh, it's a defensive cell, right? White blood cells can appear immune system make a part 10. So immunity. So how these macrophages, how they do this immunity, they eat everything, right? Whenever they see something that is bad, like a bacteria or a pathogen, like a virus or whatever, they try to eat it. Okay, macrophages always try to find solution or to protect you by eating stuff, right? Eating stuff. So to do that, they use these lysosomes. How they use these lysosomes, I tell you, right? So here is, imagine this is a bacteria. This food is a bacteria. So this white blood cell will go and take the bacteria into its inside, right? So compare. So now don't think bacteria, I mean, your white blood cells, these macrophages are actually very big compared to your typical bacteria, right? Very big, right? These are eukaryotic cells, your white blood cells. So when it encounters something that is good to eat, right? Then what is going to happen? It is taken into the cell. Then comes the weapon, that is the lysosome. This is the lysosome. The lysosome comes and combines with the food particle and will release its enzyme. So the lysosome contains enzymes, right? The lysosome will release the enzymes and it will digest whatever the thing that the cell took in, right? So that will destroy it. And also the cell will get some nutrients. So the bacteria may have some proteins, some carbohydrates, lipids. They will be used by the this big cell. Get it? That is one of the functions of lysosomes, right? To digest food particles received by phagocytosis, right? So it says here, digest food particles received by phagocytosis. Please highlight the word phagocytosis. 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 Okay. Then here it says, what are the contents of these lysosomes? Hydrolytic enzymes which catalyze the breakdown of carbohydrates, proteins, lipids, and nucleic acids. So hydrolytic means what? What is hydrolytic enzymes? Hydrolytic enzymes. They do hydrolysis. Hydrolytic enzymes do hydrolysis. Okay. How do they do hydrolysis? How do you do hydrolysis? What is hydro? You are adding water. So when you add water to a polysaccharide, what do? When you add water into the bonds of a polysaccharide, you start to break down the bonds, right? Because why? Your glycosidic bonds. I hope you remember. Glycosidic bonds are what sort of bonds? They are formed by what sort of reaction? Glycosidic bonds are formed by what sort of reaction? Dehydration. Dehydration reaction. So the opposite, the reverse of this happens. So to make a polysaccharide, you remove water, right? So to make a polysaccharide, you remove water. To break a polysaccharide, you add water. When you are making a polysaccharide, you remove water. That is dehydration, right? To break a polysaccharide into the monosaccharides, you add water. That is called, adding water is called what? Tell me. Adding water is called? Yeah. I just talked about hydrolysis. Hydrolysis is done by what? Hydrolytic enzymes, right? So this applies to proteins also. To make a peptide bond, what do? You do a dehydration reaction, right? So when you want to break a peptide bond, you add water. Get it? Okay. Then, right. So that is phagocytosis. And there's an interesting thing called, and also lysosomes, after this digestion, exocytosis is something like, so here it's the digestion is completed. No? So there are things the cell doesn't like, there are things that is not required by the cell. Cell එක වැඩ නැති like කුණු තියෙනවනේ, right? It's like fecal matter. අපි නිකන් කාලි ඉවරලා after we have digested our food, there is something that is we don't need. No? That is our fecal matter, no? Feces. It's like that. So the cell is going to the toilet, right? That is done by what? Also by the lysosome, taking out the trash. කුණු ටික එළියට අරන් යනවා. That is also done by the lysosome. Right? It is called exocytosis. 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 Then digest worn out organelles. This is such an interesting thing. Right? It's called autophagy. 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 I didn't put this in the tube because it takes some space. You can take a screenshot of this phagocytosis and autophagy. Autophagy means when an organelle, right? We'll talk about these organelles such as mitochondria and chloroplast. These are very active organelles. They are always very active. Mitochondria is always making energy. The chloroplast is always doing what? Photosynthesis. So with time, these organelles can get damaged, right? So the mitochondria has so many enzymes. These enzymes can get sort of, used up, right? These enzymes can change, right? So with time, it's like an engine, right? So if you run an engine for a long time, it's going to get worn down, right? Its parts are going to be worn down. So after some time, you have to throw out that engine. You have to throw out that mitochondria. You don't just throw out the mitochondria. You actually recycle the mitochondria. That is done by lysosomes. So this is one of these Vayasaka mitochondria, right? So they have worked very hard and now it's a damaged mitochondria, worn out mitochondria. What is it going to do? There's a lysosome coming and it's going to bind with the mitochondria and it's going to digest. And the sort of the proteins and the other components of the mitochondria will be recycled by the cell, maybe to produce a new mitochondria. We don't know. Okay. Got it. Yeah. That is called what? That is called autophagy. Autophagy. Something additional I can give you, right? Something additional, I'll explain it to you. Right? Not only these sort of macrophages, not only these sort of white blood cells. Have you heard of amoeba? Amoeba. Amoeba is a unicellular protozoa. Protozoa. They also eat by this phagocytosis. They go near a food particle and they surround it. They surround it and they take it inside. Then they digest it with lysosomes. Lysosomes. Sorry. Okay. Any questions? Lysosomes. Write down here. There's some space. No? Write down a few facts. Write down a few facts. Okay. You want me to explain worn out organelles again? Right. The organelles, if after they have functioned for a long time, especially your very active organelles, such as your mitochondria and chloroplast, they get damaged, right? Think of a chloroplast. The chloroplast, what is the action of chloroplast? It absorbs sunlight and that converts that sunlight uses that sunlight to produce energy, right? Sunlight energy will be converted to what we call as ATP. You know ATP, no? I forgot. You know ATP, no? The light energy in the sunlight will be converted to what? chemical energy. Light energy in the sunlight will be absorbed by the chloroplast. We'll be learning about the chloroplast. So your chloroplast is like this. It has inner membranes like this, right? Inner membranes like this. And there is sunlight coming. And the sunlight, it has heat energy, mainly light energy, electromagnetic energy, that will be converted to what? Converted to ATP. ATP is what sort of energy? It's chemical energy. It's chemical energy. So when this chloroplast is getting exposed to sunlight again and again and again, it's going to be damaged by the sunlight. That is one of the commonest ways, right? So with time, this chloroplast is going to be a bit useless. It's worn out. So there's no need of, it's useless to keep that. Therefore, what is going to do? You're going to recycle it. Who is going to come? A lysosome is going to come. It contains these phytolytic enzymes, and it fuses with the worn out organelle and releases its digestive enzymes. And these digestive enzymes will start to break down the organelle. Got it? Yeah? Understood? Okay. Right. Write down under lysosomes, protozoans, protozoans such as amoeba, scientific name, amoeba, protozoans such as amoeba ingest food particles by phagocytosis. Ingest food particles by phagocytosis. Ingest food particles by phagocytosis. These food particles will be digested by lysosomes. These food particles will be digested by lysosomes. All right. Protozoans like amoeba ingest food particles by phagocytosis. That's what I said, right? By phagocytosis. Then these food particles will be digested by lysosomes. Okay. Then write down, I'll explain this after writing down. Dead cells are digested. Dead cells are digested. Dead or dying cells. Dead or dying, okay? Dead/dying cells. Dead/dying cells. Dead/dying cells are recycled. Dead/dying cells are recycled from inside the cell. Dead/dying cells are recycled from inside the cell when lysosomes release when lysosomes release hydrolytic enzymes into the cytosol. When lysosomes release hydrolytic enzymes into the cytosol. This is called autolysis. This is called autolysis. Autolysis. Auto means self digestion. Autolysis. Okay. This is called autolysis. Which one? Do I need to repeat? No, right? Everyone got it? I didn't send the updated PDF. No? I didn't send it because I'll be sending the printed one. You want me to repeat it? What did I say? I don't remember. Dead or dying. Can someone put in the chat? Someone who wrote down? Put in the chat. Okay. So, autolysis. It's an important concept. So, I mean, when a cell is damaged beyond repair, there is no use of this. There is no use of keeping that cell anymore, right? When a cell is damaged beyond repair, there is no use keeping it. So what is it going to do? It's going to sort of self-destruct. It's going to suicide by releasing. So inside the cell, they will have so many lysosomes. What the cell is going to do is it's going to commit suicide by releasing all the lysosome enzymes into the cytosol, into the inside of the cell, right? Then these enzymes are sort of free inside the cell. The cell is going to start digesting from inside the cell. So it is going to cause autolysis. Now I'll explain something a bit interesting. You have this when you're doing reproduction, you will find this. There's a special lysosome in the smallest cell of your body. There's a special lysosome in the smallest cell in your body. What is the smallest cell in your body? Smallest cell in the body, and females don't have this. What is the smallest cell you have? Right? What is the smallest cell you have? What is the smallest cell you have? Anybody recognize this? Yes. This is a sperm, right? A sperm, which is the male gamete, right? Produced by the male by cell division, meiosis cell division. So it has the father's DNA in the head. So it has three parts. This is called the head. This is called the midpiece. It is sort of the body, and this is the tail. Okay. So this is a typical sperm. So when compared to the sperm, your egg is thousands of times bigger. Thousands of times bigger. So I'll draw it like this. It's like a spacecraft landing on the moon, right? It's if your egg is the moon and a sort of a small spacecraft landing there is compared to the sperm, right? So your the female egg, actually it has several protective layers, right? It has several protective layers as well as sort of protective cells around it. So inside this is the egg, okay? Ovum. Ovum. We call the egg as ovum, right? Here in the sperm's head, you have a special lysosome. A special lysosome. No, it's actually like one big lysosome, right? It's called the acrosome. Acrosome. It contains these hydrolytic enzymes. So when the sperm needs, so the sperm needs to get inside the ovum because inside you have the female DNA, right? Inside the ovum you have the female DNA. So this male DNA and the female DNA needs to combine to make what we call as a zygote. That will become the baby, right? So in order to get through these supporting protective cells as well as a thick barrier, right? It's called there's a thick barrier called the zona pellucida. All these words are in your syllabus, okay? Nothing out of syllabus. All zona pellucida, right? So this sperm has to drill through it. It has to sort of drill through the protective cells as well as these protective layers. So to do that, it has a weapon. On its head, it has a bag of enzymes that is going to help to digest through these protective cells. That is called the acrosome. Okay. Draw a small rough diagram of a sperm and label this as head. This is called midpiece. All these are in the syllabus. Okay, nothing out of syllabus. This is the tail. Draw a small sperm, and in the head, draw a small vesicle called the acrosome. Okay, acrosome. Then I will give you a small note. Okay, done. Yeah. Okay. Write down. In the head of the sperm, in the head of the sperm, you have, in the head of the sperm, you have a specialized lysosome. In the head of the sperm, you have a specialized lysosome called acrosome. Called acrosome. Called acrosome. That helps, that contains enzymes, that contains enzymes, that contains enzymes, helping the sperm, helping the sperm, helping the sperm to penetrate, helping the sperm to penetrate the protective layers of the ovum, female egg, help to penetrate the protective layers of the ovum. Okay, right. Then we move on to the peroxisome. That is sort of like the twin brother of lysosomes, but a bit more, let's say, a bit more dangerous, right? Involved with doing some bit more dangerous things than lysosomes, right? So they are very similar to your lysosomes in structure, but they have different types of enzymes. They contain what we call as oxidizing enzymes. Right? So oxidizing means they are going to oxidize something else, right? So they are going to induce oxidation. What are they mainly oxidizing? They mainly oxidize hydrogen peroxide, H2O2, right? Hydrogen peroxide, right? They mainly break down hydrogen peroxide. So they will break down hydrogen peroxide. Your hydrogen peroxide, especially this OH2 2- group in hydrogen peroxide, is very reactive. It's very dangerous, right? So we don't like high peroxides inside our cells. So we are going to break down this hydrogen peroxide into just water and oxygen gas. So one of the enzymes that is inside this peroxisome is an enzyme called catalase. Catalase. That is going to break down hydrogen peroxide into water and oxygen. Right? That is the main basic function of peroxides. Okay. So in terms of function, that is the first most important function. Then there's something called photorespiration. Okay. Photorespiration, I won't go into trying to explain it right now, because in about another month, you will know what is photorespiration, right? It's part of photosynthesis, right? It's think of it as like a wasteful process. It's like a byproduct of photosynthesis that requires the peroxisome's help, right? Peroxisome help to solve a problem that is called photorespiration. Okay, for now, no need to. I'm not going to explain that. Then what else? What are the other functions they say? Right, detoxification and photorespiration. Then specialized peroxisomes called glyoxysomes are found in fat storing tissues of plants. They convert fatty acids into sugar. This is a bit of a complicated process, okay, glyoxysomes, but it also requires oxidation of fatty acids. So it is actually called beta oxidation. This actually happens in our body as well when we are using fatty acids to get energy. But we don't have this glyoxysomes. Okay, it's an oxidizing process that is going to convert fatty acids into sugars. Very, very important. It is only found in glyoxysomes are only found in plants. Very, very important. Glyoxysomes are only found in plants. Okay. So in your tute, underneath where you wrote a few things about the lysosomes, write down, write down. Write down. Write down this reaction. That is enough. H2O2 break down by catalase into water and oxygen. That is enough. Write this reaction down. Put the small topic peroxisomes and write down this reaction. Then write down under that. Write down under that. Write down under that. Peroxisomes are found in both plants and animals. Peroxisomes are found in both plants and animals. Peroxisomes are found in only both plants and animals. Plant and animal cells, okay? That's what I mean. Peroxisomes are found in both animal and plant cells. Animal and plant cells. But glyoxysomes, the word is in your resource. Glyoxysomes. Glyoxysomes are found only in fat storing tissues of plants. Fat storing tissues of plants. Okay. Right. So I just want to ask, how have you found, have you got yourself a good copy of the resource book? Right? Do everyone have a good copy of the resource book? Everyone has, right? How did you get it? Did you get it printed out, or I want to know? මොකක්ද කරේ resource එක ගැන? Resource book එක ගැන? Right? Because many bio sort of teachers, they sort of give the resource book as a printout. But I think that's kind of illegal, right? You are not allowed to reproduce. You are not allowed to reproduce the resource book for commercial purposes. You're not allowed to sell the resource book, right? I think some of the shops do actually. But anyway, I'm not going to give you the resource book, right? It's not, even if, especially for an online class, you don't need. So, have you got it? Everyone has, right? Hands up, anyone who doesn't have the resource book? No one, right? If you, you can buy from the NIE, it's kind of cheap. But if you buy it, I sort of recommend that you sort of remove the cover, right? Take out the cover and sort of spiral bind it. හරියට දීලා සම්පූර්ණයෙන්ම cover එක ගලවලා pages ටික ඔක්කොම separate කරලා do a spiral bind. Otherwise, all the pages are going to fall out, right? So that's going to make you sort of hate the resource book when all the pages are falling out. Okay. So invest. Yeah, you bought from the NIE. Try to do that. Oh, if you can protect the resource book, then it's fine. But I sort of recommend that you take the resource book everywhere you go. Bus එක යනකොට හරි trip එකක් යනකොට හරි you take it. At least a soft copy you take it. Right? At least take a soft copy and just go through it, highlight it, right? Keep it, sort of treasure it, right? Think of it as a prized possession that you cannot pass, that without it you cannot get an A, right? Think of it as a prized possession. There are so many flaws with it. There are so many things wrong in the resource book. But anyway, the system is now, without the resource, you cannot get an A or even pass. Okay. තව මොනවද? When are your results coming? All your results in the When are your results coming? Next month or this month? It's been so long, no? Since you did O-levels? Next week? Results are coming next week. Okay. Right. So I hope everyone will continue doing bio, right? There's no such risk, ne? No one has such a risk, ne? Dropping out of bio, right? I mean, yeah, all over result doesn't matter except for the important subjects. Yeah, science, maths, English. Those are the actually only one that matters, right? So anyway, so after 15th, then you are getting results, no? So I hope that will be sort of after that point we'll be sort of picking up the pace, right? Because this, I mean, this is like a transitioning period, no? So more students will decide to do bio, and some students who are not sort of doubtful, they will sort of drop out. I hope nobody here will do that, right? Anyway, so from after your results, this is going to be proper A levels, okay? Proper A levels, and not just this sort of introductory stuff and like that, right? So be prepared. So now you have your vacation. You just come to class and just listen. So after that point, you sort of have to start getting into the A-level rhythm, okay? Okay, right. Peroxisomes are done. Peroxisomes are done. Any questions? We did Golgi, lysosomes, peroxisomes. Today we are going to do mitochondria and chloroplast as well. Now we come to sort of complicated organelles. These mitochondria and chloroplast after the nucleus are kind of the most complicated organelles. Right? They are very complicated. They are sort of their own cells. Their own cells. They are sort of like their own cells inside of a cell. They act as separate cells, but inside of another cell. You will know, you will understand why. There's a theory of evolution, cell evolution, called endosymbiont theory. Endosymbiont theory. This theory states that, so in your third lesson, that is evolution and diversity, you will understand how in our early oceans, the first cells were formed. In the early oceans, our first cells were formed, right? So there's a theory when the first eukaryotes. So the first cells to occur are called your prokaryotes. Your prokaryotic cells first originated, right? We now believe that the life originated here on Earth, right? Asteroid dating our වෙන්නත් පුළුවන්. But there is evidence to say that the first cells, therefore the life, originated alone. I mean, without any external influence in the earth by itself, right? In the earth by itself, in the early oceans, to be more exact, in the early oceans. So the first cells were prokaryotic cells. So after about billions, I mean about 2 billion years later, they got eukaryotic cells. So after evolution, they got eukaryotic cells. So with the origin of eukaryotic cells, there's something interesting happens. So it's still a theory, but there's so much evidence to that. It is called this endosymbiont theory. So I'll screen share. So when the first eukaryotic cells originated, there's a theory that they engulfed, they ate some of the prokaryotic cells. This is the endosymbiotic theory. So here is an early eukaryote. We call it a proto-eukaryote. Proto means what? It's like not a proper eukaryote, but a kind of a, let's say, like a trailer version of a eukaryote. It's not a proper eukaryote yet, but it's going to be a eukaryote. So it's a proto-eukaryote. So what is happened? Infolding. So this step is not necessary. It says how endoplasmic reticulums are formed. This is the important thing. So this explains how eukaryotic cells got mitochondria. How did eukaryotic cells get mitochondria? They consumed an aerobic bacteria, right? So this eukaryotic cell ate a aerobic bacteria. What is aerobic? What is aerobic? Aerobic means produce energy with oxygen, with use of oxygen. Okay. Aerobic bacteria. So this early eukaryotic cell ate a aerobic bacteria, and that bacteria inside the eukaryotic cell became what? It became a mitochondrion. It became a mitochondrion. You will see how we can prove this. There is so much, let's say, evidence. Then the next thing, in a second event, this eukaryote consumed a photosynthetic bacteria as well, right? Right? Because it's a big, this eukaryote is a big cell, it can engulf this. So it engulfed a photosynthetic bacteria as well. So that photosynthetic bacteria became what? With time, it became what? Any guesses? With time, it became what? Yes. Come on.